All right, so last lecture for this semester, November 22nd, Nucleic Acids Part 2. So last time you got all the way up into tRNA and how that works, and then you should have watched a short YouTube video about what that process looks like in action. So sometimes that's really helpful to see. And so we talked about pretty much up to transcription. We replicated our DNA. Then we DNA transcription to RNA. And then we're going to do the third step, which is the translate from RNA to proteins. Okay, so that's today. And then we're going to talk about mutations and viruses. So the genetic code is the identification of each group of 3 nucleotides and its particular amino acid. So we're going to have 3 nucleotide bases, which will equal 1 amino acid, and then that will continue on and on and on and on until we get the entire sequence for the protein from the RNA. Okay. So translation is what we call this process. It's how we take the information encoded in mRNA and use it to direct the sequencing of the amino acids to make the functional protein. Okay, so we talked a little bit about this when you watched the video last time, so I'm not going to reiterate it. I just want to point out the amino acid is coordinated to the tRNA through a bond, and that's the purpose of the tRNA, is to bring it to the place to where it needs to be so that it can be synthesized, the entire amino acid. So the important part is, how do we go from mRNA nucleotide bases to protein amino acids? And the way we do that is this little chart: the first base, the second base, the third base. So for example, say I have UCA. So the way this would work is I'm going to go to the U box. I'm going to go to the row. I'm going to go all the way over to where the second base is my C. And then I'm going to go down until I get to the third base, which is an A. Oops, I drew my arrow on top of it. How about that? UCA. Okay? So my nucleotide uracil, cytosine, adenine, in that order, codes for the amino acid serine. Okay? And so all of them will code for an amino acid, and you'll see that there's a lot of repetition. There are just a few examples of where we have what's called a stop code. And so UAA, UAG, and UGA all will cause the stop. And so usually these stop codes are at the end of the sequence. Without the stop code, it'll just theoretically go on and on and on and on. So the stop code is necessary to stop and then release the protein so that it can go on its way to do its job. Okay, so let's practice a question. I think I put it in the wrong order though. I did. So here is our in-class, I believe this is our second to last in-class question. Again, it's still September when I'm recording these, so my brain is not quite sure. But it should be November 22nd in class. And we are going to do number 2, number 6, probably need some more space. Number 2, number 6, and number 8. This should help you with your sapling homework as well. So the first one says to write the codon on the mRNA that would pair with the tRNA, and so it's the complementary base pairing. The only difference is that the 3' and the 5' are going to match up like that because they're opposite. They're unzippering in different directions, if you will. So one is being read this way, the other one's being read that way. So the way it works is I'm going to start with 3', and then I just match my A's and my U's, my C's and my G's, and that's it. So that's A, If we do B, it's 3 prime C U U, because again this is mRNA, or we're talking about RNA, not DNA, and then 5. Alright, so take just a few seconds and do C and D. And see if you can write out the correct code. Alright, so you should have gotten A, G, G, and G, U, G. Okay, so hopefully you got those as your answers to 2C and 2D. All right. Next one, number 6, says use figure 15.4 to identify the amino acids carried by each tRNA molecule from exercise 2. So the one we just did. So now we need to take our 2A, 2B, 2C, 2D, and then look for them on the chart. So I have AAC. So let me erase what I did before. So I go down to A, and I'm looking for AAC. So I believe this one is oh wait, we're looking at the wrong one. We need to use the mRNA that's right here. Okay, so we need to use the UUG. Okay, because the codon is going to code for the amino acid. So UUG, UUG. So this is going to be leucine. Okay, I'm not going to expect you to know the 3-code sequence. I'm not going to expect you to memorize that chart, but I definitely don't expect you to memorize the abbreviations for the amino acids. So GAA is our next one. So GAA is glutamine. Okay, so now our next ones are UCC and CAC. So I'm going to write them up here, and I want you to practice using the chart. And you should have a copy of this chart along with the copy of the amino acids. So you should have 2 handouts for this test. And if I asked you to do this, I would give this to you on the test. It would be given. Okay. Okay, so let's see. If I start off with the first one, I have first base is U, second base is C, so I'm looking in this box right here, and UCC is serine. Okay, so my next one is CAC, so first base is C, second base is A, so I'm looking in this box right here, and CAC is histidine. Okay, so let's go down, and remember you use the mRNA code to determine the amino acid it codes for. Sorry, number 8. Use the figure in 15.14 to determine the amino acid sequence produced from this mRNA sequence. Okay, so we are going to need to break it into groups of 3, okay, because it's 3 code sequence. So we are going to have AUG, GCA, AUC, CUC, AAA, CGU, and UGU. Okay. So now you guys have an advantage that you have your table in front of you. So I'm going to have to go up here and use this one. So if we're looking at the first one is AUG. So I have AUG. So that's methionine, MET. So we're going to write it out, MET. The next one is GCA. So I have GCA, that's alanine. So I'm just rewriting those 3 letters to represent the amino acid. My next one is AUC. So now I have AUC, isoleucine. So it's ILE. The next one is CUC. So I have CUC. So this one is leucine, LEU. Now we're going to have AAA. So we're going to have AAA, which is lysine. LYS. CGU. CGU. So arginine. ARG. And the last one is UGU. And let's see, UGU. So UGU. So cysteine. Okay, so this would be our amino acid sequence, and normally what Sapling's going to want you to do is MET, ALN, ILE, LEU. So you can see I'm just putting them out in order, and I'm putting dashes between them. Okay? So that's how you go from the mRNA to the Anderson, please don't. In order, how you go from mRNA to amino acids. Okay? All right, so let's set this aside. We have one more after we talk about mutations. So mutations and genetic diseases, this is often the manifestation of some sort of chemical or physical change that alters the DNA, the nucleotide sequence specifically. Okay, so one example is called a point mutation, which is a change in which one nucleotide is substituted, added, or deleted. Okay, so it's the change of one nucleotide. But one nucleotide being deleted, it can cause a huge problem upon expression of that eventual protein. So a mutagen is a chemical or physical agent that causes mutations. So we can think of lots of examples. We often worry about chemicals that cause cancer, but they also can cause mutations. And so a genetic disease is a hereditary condition caused by an altered DNA sequence. Because it's altered and it's in the DNA, it can be passed on. All right. So what do these look like? Well, let's say that this is your normal sequence right here. If we have a substitution, you can see that the AAAATG, the one that's being substituted, is instead of the DNA having a cytosine, it has a thymine. And what happens is when the mRNA comes along to copy, it's going to have an A as the nucleotide base, which then will eventually code on to be lysine and not glutamate. And this is called substitution. You're substituting one for another, but it results in a change in the amino acid. Okay, this changes, or could change, protein function. All right, the next one is an insertion, and this is where instead of there being the CTC up here at the top, an extra cytosine is inserted into the sequencing, and it changes everything downstream. This now becomes a glycine, and this becomes arginine, which wasn't either of the amino acids that were there before. Deletion is the exact opposite. Instead of inserting one, we are deleting one. In this case, the A was deleted from the original sequence, and it changed all 3 amino acids that resulted, even though these 2 are now the same, which is interesting. Okay, now here's the thing about that chart. So let's go back up to this chart, and let's look at how much repetition is present. Isoleucine is present 3 times. We have histidine, which is several times proline, serine, leucine, valine, alanine. So the repetition in this chart is what prevents every single change in the nucleotide sequence from resulting in something that's deathly. Because there's repetition, it means that if we have a first and a second base that's cysteine, it doesn't matter if the third base is any of the other nucleotides, it's always going to code for proline. So this is the way that the genetic code protects itself from having devastating mutations every time there is a point mutation. Okay, so it's a really beautiful, wonderful thing if you think about it, that we can have a small change in the code that doesn't change anything overall, and it's what protects the genetic mutations from basically resulting in a species that would not survive due to hereditary issues. All right, so now we're going to practice identifying the mutations. So it says a portion of the coding strand was found to have the sequence of here. A mutation occurred in the gene making the sequence this. Identify the mutation as a substitution, insertion, or deletion. What effect would the mutation have on the sequence? So we're looking if there's any change. So we have ATG, ATG, GC, AA, TCC, TC, AA. Okay, here. So here's where the change occurs because the A and the C don't match up. But if I go on to look, the T, G, T, C, G, C match up. So it is the A, the adenine, that has been deleted. Okay? So in order to answer that question, we have to compare what we had to what we have now. Okay? All right. So now it wants us to say, would the mutation have an effect on the amino acid sequence? So we have to look at the base pairing. Now the nice thing is nothing is going to change until we get to this deleted area. Okay. So we have to look at, does AAA and AAC code for the same thing? So AAA, AAC. So let's go back up to the top. Not to the top, but to our chart. And now we have, excuse me, A, A, C, and AAA. So we do have a change in the amino acid sequence. We don't have to look any further. We can see that asparagine and lysine are not the same amino acids. So this answer to this question is yes, it would change the sequence, change the amino acid sequence. Okay, and that could be enough. It's impossible to say if it definitely would result in a genetic mutation that was physically manifested, but it is enough to change the amino acid sequence, which could either render the protein inactive or harmful. All right, So the last little bit of our topic is going to be about viruses. And so we are usually familiar with viruses because if you get a cold or anything like that, a viral infection, we have heard that before. And we compare that usually to something that's bacterial. So bacterial viruses, things like that, you usually get antibiotics. When it's viral, we usually have to let the immune system do its job. And sometimes we have immunocompromised people that get very, very sick. Otherwise, if you have a healthy, innate, and acquired immune system, you should be able to fight most viruses. So, a virus is an infectious agent that is much smaller and simpler than bacteria. Okay, so it makes it sound like it's not that bad, but we do know that there are examples of viruses which are very, very devastating. So, a retrovirus is a specific type of RNA virus that directs the synthesis of a DNA copy in the host cell. So it's kind of an invader, a sneaky one, a smart one. Okay, so just because viruses are smaller and simpler does not mean that they are not dangerous. Okay, so these are some examples. I love how these look like little robots of different viruses. Okay, so we have different shapes. This one kind of looks like what you would use to inflate like a workout, an ab ball. I don't know why, but that's what it looks like to me. Okay, but you can see here they have kind of their own nuclear information there in the center. Okay, so the most common type of retrovirus that we know about is whoa, so sorry guys, my split screen. The one that we know the most about is the HIV, right? That eventually will cause AIDS, and that's the best known retrovirus. But the definition is kind of cut off here. So a retrovirus is an RNA virus that directs the synthesis of a DNA copy in the host cell. Okay, which we have the definition up here, but I want to have it here on this slide. And so if we look at what that looks like, we have the virus here, the host cell. Once the virus binds to the host cell, you see here it's kind of injecting these little green things, which is viral DNA. So the viral RNA is inserted, reverse transcription of viral RNA to form DNA. Okay, so now we have viral DNA in our cell. What happens is this is integrated into the host genome, and then the cell with viral DNA is incorporated. And then what happens is the expression of the viral DNA is now viral RNA, and then it goes back out. Okay, and it replicates. So at this point now, this is how it takes advantage of the host cell's inability to necessarily fight off this type of approach. Okay, so it doesn't attack your immune system in what I would say is the exact same way. So HIV affects approximately 33 million people worldwide, but because of modifications to medicines and advancements, not everybody who gets HIV will necessarily develop AIDS. And AIDS is the one that really, in the end, your body can no longer fight. You normally will pass away of what's called ARC, so AIDS-related causes that are because of the devastation that AIDS causes to your body. So on a lighter note, I think there are some advances that are making these viruses not so devastating, but we still are fighting an uphill battle when it comes to bacterial infections because antibiotics are being used for everything. Every time someone gets a sniffle and they suspect it could be bacterial, we prescribe antibiotics, and so your body becomes immune to that, and the viruses, the bacterial infections, they become smarter. And people don't finish their antibiotics. They take until they feel better, and they save the rest for later, another rainy day. And so while we're making advances on how to treat viruses, bacterial infections are going to be kind of problematic as well. So a lot of conceptual stuff at the very end. My hope was that this lecture would be short enough. I don't think I turned my timer on. Oh, I did. So this should have been about 20 or so minutes. And so my hope is that you have lots of time to ask about chapter 15 questions to ask about exam 4. You should have had a power hour hopefully before now, because your exam 4 is on Monday before Thanksgiving. And then just a reminder that your final exam, which is cumulative, it's only going to be for exams 1 through 3 though, okay, because you're not going to get exam 4 back in time to be retested on that material. So your final exam is exams 1 through 3, and the test is on Wednesday, December 4th at 10:30 a.m. And you have 2 hours to take the test. I suggest going over material, looking at your previous exams, and you should have them back now, because I left them in the office for you guys so that y'all could have exams 1 through 3. Answer keys will also be available to you on Canvas. So you should have everything you need to prepare, and then make sure you go back and look over old practice exams to prepare as well. I hope that all is well, and I missed finishing out the semester with you guys, but I hope to see you around campus in the future, and I hope you have a Merry Christmas and a Happy New Year.